The fetal pig, external anatomy, abdomen, thorax, and blood vessels. The fetal or unborn pig is the mammal of choice for dissection because it has all the basic and anatomical features common to the higher mammals, including the human, and it is easier to dissect and more readily available than other good specimens, rabbit and rat. These pigs are a byproduct of hogs that are slaughtered for food. Keep in mind that the pig you are studying is an unborn mammal and as such will have characteristics that are limited to the unborn and are lost or modified at birth or shortly thereafter. An obvious structure of this kind is the umbilical cord. You will see other temporary structures as you dissect. Along with the study of macroscopic anatomy or gross anatomy, you will study the microscopic anatomy or histology of the organs and tissues that you dissect. The slides used for histology are from various vertebrate animals. Although they are not identical with the tissues of the pig, they are nevertheless characteristic of vertebrates in general. Part A, the general external anatomy. Note the primary divisions of the body: the head, neck, trunk, tail, forelimbs, and hind limbs. Compare these divisions with those of your own body. Probably you have already identified the cord extending from the middle of the underbody as the umbilical cord, with which the fetus was connected to the placenta in the uterus of the mother. Look at the demonstration showing this relationship. At birth, the mother bites off most of the umbilical cord and the rest degenerates. The healing over of the spot where the umbilical cord joined your own body left a scar, the umbilicus, or navel. Now examine the body of the pig carefully, region by region. The head includes the mouth and jaws, the nostrils and the snout with its dorsal elevation used in brooding, the eyes whose lids are fused together in the smaller specimens, and the external ears with the auditory tubes leading to the eardrum. In the pig, the neck is so thick as to be nearly obscured. This thickness is caused by the great development of the muscles used in rooting. The thorax, or chest, is the part of the body with the ribs, forelimbs, and shoulders. Note the division of the forelimb into the upper leg, lower leg, wrist, foot, and toes or digits, and their partial correspondence with your own arms. This correspondence becomes more evident if you compare the skeletons of pig and man. In the pig, only 2 digits of the ordinary 5, the third and fourth, are fully developed. The second and fifth are reduced, and the first is completely lost. Feel for the ribs and find the posterior border of the thorax. This bony box encloses the lungs, heart, and major blood vessels. The abdomen is the part of the body with a soft underwall from which the umbilical cord extends. Note the 2 rows of teats or nipples on either side of the cord. Within is the abdominal cavity containing the stomach, intestine, and kidneys and other viscera. The sacral region includes the hind limbs, pelvic bones, and their attachments to the sacrum. Compare the hind limbs with the forelimbs and with your own. Find the anus, the posterior opening of the digestive tract just below the tail. In the female pig, the vulva, in which openings of the reproductive and urinary tracts occur, is found just below the anus. In the male, the external opening of the penis may be found in the ventral midline, posterior to the umbilical cord. 2 swellings mark the scrotal sacs on either side of the midline, just below the anus. In the caudal region, the tail is small in the pig. It is even smaller in the human, being reduced to the coccyx, a structure seen in the skeleton on display as a short extension of the vertebral column. Part B, the abdominal region. Number 1, umbilical structures. Find the white cord in the abdominal cavity extending anteriorly from the umbilical cord. This is the umbilical vein, which drains the fetal blood from the placenta and into the liver. You may sever this vein, but keep its position in mind so as to be able to trace it later. 3 cords will be visible beneath the flap of the body wall, now continuous with the umbilical cord. The lateral pair are the umbilical arteries, through which the fetal blood flows to the placenta. The larger, hard-walled sac in the center is the urinary bladder. The allantoic duct carrying the nitrogenous wastes from the bladder runs through the umbilical cord to the placenta. The fetus thus receives oxygen and food from the mother through the placental circulation, and also unloads carbon dioxide and nitrogenous wastes there. The abdominal viscera make a pair of lateral incisions through the body wall. Examine the viscera, the soft internal organs, separating them with your fingers, being careful not to tear anything. Most obvious are the loosely coiled small intestine and the thicker, tightly coiled large intestine. They are supported by a thin sheet of tissue, the mesentery, which suspends all of the intestines from the mid-dorsal body wall. The mesentery is formed of two layers of tissue, one continuous with the lining of the abdominal cavity on one side, and the other layer continuous with the lining on the other side. The lining of the abdominal cavity is called the peritoneum. Find the soft, white-walled stomach anterior to the intestines, with the dark-colored spleen along its left posterior border. A light-colored, roughly granular structure, the pancreas, is found in the mesentery between the stomach and the first portion of the small intestine, the duodenum. Anterior to the stomach is the dark liver. How many lobes does the liver have? Note in one of the liver lobes a dark green sac, the gallbladder. It is green because of its contents, the bile. Its duct, the bile duct, drains into the duodenum. The pancreatic duct, less easily traced, opens into the duodenum at about the same place. Now shove the intestines forward, and find the posterior continuation of the large intestine, the rectum. It is partly concealed by the urinary and reproductive ducts, which will be studied later. Behind the peritoneal lining of the dorsal part of the abdominal cavity are the relatively large, hard kidneys. Cut the peritoneum along the lateral border of the left kidney and peel it off towards the midline. Look for the left adrenal gland, a crescent-shaped body under the peritoneum, immediately in front of and closely applied to the kidney. In life, the adrenal glands are orange in color, but in death they are brown because of the large amount of coagulated blood they contain. Finally, explore the diaphragm, a membrane, thin in the center but thick with muscles around the edge, which separates the abdominal and thoracic cavities. Examine the demonstration specimens of the dogfish shark and bullfrog and compare the abdominal viscera with those of your pig. Part C, thoracic region. The glands. Note two large masses of white granular tissue, concealing part of the heart and interior blood vessels. These are the thymus glands, which are comparatively large in young animals, but degenerate after sexual maturity. They extend well into the throat region, where they must be distinguished from muscles, particularly those which move the voice box or larynx. Another gland you will find in the throat region is the relatively small oval thyroid gland, flat against the trachea just in front of the thorax. Normally reddish, the gland may be cream-colored or brown in the preserved specimen. Also find the salivary glands. Pull the skin farther away from the left side of the head up to the eye and behind the ear. Two of the glands, the submaxillary and sublingual, are found close together just under the posterior angle of the jaw. These are compact and resemble the thymus. The third salivary gland, called the parotid, is very diffuse in structure and less easily found. It is located below and behind the ear, usually plastered tightly to the skin. Examine the heart. Find a pair of thick white strands running along each side of the pericardial sac. These are the phrenic nerves. Remove the pericardial sac from around the heart, being careful not to sever any blood vessels or nerves. Note that the pericardial sac is firmly attached only where the blood vessels enter or leave the heart. The heart has four parts, the two air-like auricles at the front and sides, and the fused ventricles in the center and posterior. A diagonal line, marked by branches of a coronary artery and coronary vein, passes across the ventral surface and separates the borders of right and left ventricles. Heart, respiratory system, and digestive system. This exercise considers the mammalian circulatory, respiratory, and digestive systems as shown in the fetal pig. It assumes you have already begun the dissection of the pig. You will be expected to be able to trace the course of the blood from the heart to any part of the body and back to the heart, and eventually to understand the major chemical changes that occur in the blood in the various capillary beds. Sever the precaval vein where it enters the right auricle. Turn the heart forward, find the postcaval vein and cut it about half an inch from the auricle. Find the pulmonary veins from the left lung and trace their entry into the left auricle. Cut them, at the same time exposing the left pulmonary artery. Cut it at the surface of the lung. Only the aorta and its branches now remain intact. Sever the brachiocephalic and left subclavian arteries between the aorta and their first branches. Cut the aortic arch about an inch beyond the left subclavian artery, and remove the heart from the body. Examine and put it back once or twice to keep its proper orientation in mind. Go through the following steps in dissecting the heart, tracing in sequence the course of the blood through the heart and lungs. You may have to rinse or pick out coagulated blood from some of the chambers. Refer constantly to Figure 1, which shows where to make the successive cuts. The summary of the circulatory system. The circulation in the adult mammal is divided into two separate major courses. A, the systemic, arteries, capillaries, and veins to and from all body tissues, including the coronary circulation of the heart itself. And B, the pulmonary, which is through the lungs. In the fetus, two shortcuts are available to bypass the non-functional pulmonary circulation. The respiratory system and its operation. In breathing, air taken into the nostrils passes through the nasal passages and into the pharynx at the back of the mouth cavity. Here the alimentary, food, and respiratory passages cross. The air goes ventrally into the larynx and the food passes dorsally into the esophagus. Now study this relationship. The oral cavity. Insert your scalpel successively into the corners of the mouth and cut directly back to the jaw bones on each side. This severs the muscles that hold the jaw closed. Open the mouth and inspect the oral cavity and tongue. The details of the oral cavity can be seen in Figure 3. Cut completely through the lower jaw and tongue, in the midline anterior to posterior and ventral to dorsal, so as to separate them into equal halves left and right. Separate the halves of the jaw. Note that the jaw bones have not yet fused in front, and examine the posterior region of the pharynx. Find the epiglottis, a flap of tissue attached to the ventral side of the pharynx, and surrounding the glottis, the opening of the larynx into the pharynx. On your specimen, the epiglottis might be inserted into the posterior openings of the nasal passages. The trachea. Continue the mid-ventral cut into the larynx and trachea. Determine the relationship of the larynx to the nasal passage and the esophagus. A flexible probe or wire may be inserted into the posterior opening of the nasal passage on one side and pushed to a point where it may be seen in the nostril on that side. This passage will be dissected later. Expose the trachea back into the thorax as far as the right lung. Slit the trachea a short distance. Note the successive rings of cartilage that reinforce its walls. Cut the bronchial tubes, which connect the trachea to the lung, close to the lung. Now remember the right lung from the chest cavity by severing any remaining mesenteries. Study at the root of the lung the cut ends of the pulmonary veins and arteries, and bronchial tubes for thickness of wall and diameter of the passages. Note that the small median lobe of the lung, which partially surrounded the postcaval vein, actually is a part of the right lung. Granting that the left lung is supplied in a manner comparable with the right, consider the mechanical factors operating in external respiration or breathing. The thorax is a closed box. It is enlarged by a contraction of intercostal muscles between the ribs. The ribs directed towards the posterior at rest move together, forward, and outward when the intercostal muscles contract, thus enlarging the chest capacity. Expand your own chest and visualize how this process operates. Contraction of the muscles of the diaphragm. At rest, the diaphragm is dome-shaped and bulges into the thoracic cavity. On contraction, it flattens, the dome disappears, and the chest cavity increases in volume. This results in a decreased air pressure in the lungs, so that air rushes into the lungs from the outside to equalize the pressure outside and inside. Air is said to be inhaled. The exhalation, or breathing out, is also accompanied by two complementary mechanisms. 1. Gravity assists in the escape of air from the lungs at relaxation of the intercostal and diaphragm muscles by pulling the ribs down and thus decreasing the chest volume. And 2. Contraction of the abdominal muscles and certain chest muscles other than the intercostals force the diaphragm upward and the thoracic cage downward. Swift action of these muscles produces coughing. Exhale air rapidly once to feel the chest constrict and the abdominal muscles become taut. Tissues of respiratory and circulatory systems. Many tissues take part in the operation of the respiratory and circulatory systems. Arteries, capillaries, and veins are basic examples. Study the other related tissues on demonstration. Skeletal striated involuntary muscle is subject to conscious control. Skeletal muscles include those used in breathing and those used generally in body movement. Examine both longitudinal and cross sections for details. Cardiac muscle, striated but involuntary, is subject to unconscious control and is found only in the heart. Cardiac muscle propels the blood through the vessels of the circulatory system. Compare and contrast its structure with that of voluntary muscle. Smooth muscle is non-striated and involuntary. It is the walls of blood vessels, the digestive tract, and elsewhere. The fluid content of the circulatory system is blood. Another fluid component is the intercellular fluid, called lymph. Red corpuscles are the most numerous cell type in the blood. They are disc-shaped cells without nuclei. They contain hemoglobin, the red pigmented substance that acts as a carrier of oxygen and carbon dioxide for respiratory exchange. White corpuscles lack color in living condition. They contain nuclei that stain varying degrees of blue and are classified on the basis of nuclear form and granular inclusions. Striated skeletal muscle appears like two parallel veins per segment running along. The cardiac or heart muscle looks like strands of branches with notches in them. Smooth involuntary muscle looks like waves with dots. Neutrophils constitute 55 to 65% of the human leukocytes. The nucleus usually has 2 to 5 lobes, and the small cytoplasmic granules stain a pale neutral shade. Neutrophils are the most actively amoeboid of the leukocytes, and readily move out of the vessels into the tissues, where they perform phagocytosis. They form a large proportion of the pus at the site of an infection. They live an average of only 12 hours. Lymphocytes constitute 25 to 33% of the human leukocytes. 3 types of lymphocytes are recognized: large, medium-sized, and small. The small, shown in Figure 5, is most easily identified. The nucleus stains a deep blue, about which the cytoplasm appears as a narrow blue rim. An abnormal increase in lymphocytes occurs in chronic infections. Monocytes constitute 3 to 9% of human leukocytes. The large nucleus is pale and stringy and indented in old cells. The abundant cytoplasm is gray or muddy blue. An abnormal monocyte number is found in cases of malignancy and infection. Eosinophils constitute 1 to 3% of human leukocytes. The granules are large and red, obscuring the usually bilobed muscles. These cells usually show an abnormal increase in numbers in allergic reactions. Basophils constitute less than 1% of the human leukocytes. The nucleus and bilobed and the coarse granules stain blue. Basophils increase in number when inflammation is present. Blood platelets are tiny cell fragments without nuclei. They produce enzymes that initiate blood clotting. The lung is a tissue, and it's easier to understand from demonstrations. Note the air sacs and the walls of capillaries between them. The digestive system and respiratory system are the two that provide for intake of substances into the body. They are correlated in that the processes of digestion prepare food for absorption and use, and internal respiration provides for the oxidation of food with the release of energy. In addition to energy release, our foods also provide for growth and repair. The stomach. Again, examine the region of the pharynx for the origin of the esophagus and trace it posteriorly. Cut through the diaphragm to the point where the esophagus joins the stomach. Slit the stomach through the outer edge of the greater curvature and look inside. The larger part is known as the cardiac portion, and the smaller end as the pyloric portion. Is there any clear line of demarcation between the two parts? Continue the slit through the wall of the esophagus at one end and through the pyloric valve and the wall of the duodenum at the other. Note the heavy muscle in the wall and the pyloric valve. This is called the pyloric sphincter. Intestines. The first 2 or 3 inches of intestine is the duodenum. What relation does it bear to the pancreas? Remove the small intestine, beginning with the duodenum, by cutting the mesentery and gradually unwinding the entire structure. How long is it in your specimen? It is about 20 feet long in the human. About half of this length is jejunum, in which digestion is completed. The remainder is ileum, in which most of the absorption of food takes place. Cut open a portion of the ileum and examine the lining. Note that the lining forms a series of macroscopic folds. Many small finger-like projections, barely visible to the naked eye, cover the internal surface. These are called villi. The net result of these folds and villi is to increase the surface area of the small intestine. The liver, pancreas, and spleen. The liver and pancreas arise from the developing digestive tract in the early embryo. The spleen acts primarily as a storage center for blood and a generator of white corpuscles. The pancreas acts in part as a digestive gland, secreting pancreatic juice into the duodenum, while a primary function of the liver is that of food storage. Dissection of the fetal pig, excretory, reproductive, and nervous systems. This exercise considers the mammalian urinary, reproductive, and nervous systems as shown in the fetal pigs. The excretory system. As food and oxygen are used in metabolism, waste products necessarily are produced. These are mostly carbon dioxide, water, and nitrogenous wastes. The carbon dioxide and part of the water escape from the lungs in external respiration, and the rest of the water and the nitrogenous wastes are removed by the kidneys and pass out as urine. The kidneys, urinary bladder, and their associated ducts are called the excretory system, and the processes of waste removal are called excretion. The blood supply to each kidney contains a small artery and a pair of veins, connected by a host of capillaries. Obviously, only a relatively small portion of the total blood in the body can pass through the kidneys in any limited time. Consequently, the kidneys can remove only a small part of the total nitrogenous waste present in the blood. They act as balers, continuously removing wastes from the blood so as to keep their concentrations within tolerable limits. The kidney. Remove the left kidney and cut it lengthwise into equal halves. The center is occupied by blood vessels and a relatively large reservoir, in which the urine first collects. The layer that appears in gross section to have many fine striations perpendicular to the surface contains myriads of excretory tubules, which lead to the reservoir from tiny knots of capillaries called glomeruli. The reservoir empties into the urinary bladder through the ureter. This tube runs posteriorly to the urinary bladder. Expose, though do not injure any discrete structures that cross over it. These include the umbilical arteries and the reproductive ducts in both sexes, and the free-swinging ovaries just behind the kidneys in the female. The bladder, in turn, empties to the outside through another duct, the urethra, which will be observed along with the dissection of the reproductive system. During fetal life, however, waste pass out the allantoic duct to the placenta. The reproductive system. Normally, metabolism in the young animal leads to growth, maturation, and eventually to reproduction. Higher animals have 2 sexes in which the reproductive organs have essentially similar origin, but finally different structure and function. Because the female reproductive system is less changed from the basic pattern than that of the male, it will be considered first. In both the male and female, the reproductive system contains the sex glands or gonads, their ducts, and certain associated glands. The female reproductive system. The female gonads or ovaries are found between the kidneys, swinging rather freely from a pair of thin mesenteries that traverse the kidneys. A tube, very fine in the fetus, is coiled lateral to each ovary. This part of the oviduct is called the uterine, the fallopian tube. The oviduct continues on each side as a slightly larger tube, one of a pair of horns of the uterus. The young develop into the uterus, which becomes vastly enlarged at maturity and pregnancy. The two horns of the uterus unite into the short single body. In the human, the uterine tubes enter a single unpaired uterus. Now cut dorsally from the ventral midline until the legs may be separated widely. The cut will pass between muscles and through or between the pelvic bones, actually cartilages at this state of development. 3 tubes will be exposed. They are, in sequence from ventral to dorsal, the urethra, reproductive tract, and rectum. The urethra and the reproductive tract unite about half an inch internal to the vulva, forming a common chamber, the vestibule. The tube connecting the vestibule and the rest of the body of the uterus is the vagina. Open the vestibule and vagina along one side to see the openings of the urethra and constriction of the vagina at the base of the uterus. Fully expose the rectum and slit it open from anus anteriorly. The male reproductive system. The position of the male gonads or testes depends on the age of the fetus. The testes begin development in the same location as that occupied by the ovaries, but gradually descend into the scrotal sacs before birth. They may be found anywhere along this path in pigs of the sizes ordinarily dissected. In all specimens, the sperm ducts may be looping over the umbilical arteries and the ureters to unite dorsal to the urinary bladder. Externally, they penetrate the abdominal wall a little to each side of the midline, by way of the inguinal canals. Cut through the skin of one of the scrotal sacs and extend the incision to the point where the sperm duct leaves the abdominal cavity. The inguinal canal thus will be opened and the testes exposed. Open the sac surrounding it. The much-coiled tubule looping around the testes is the epididymis, which empties posteriorly into the single sperm duct. Cut through the skin slightly to one side of the ventral midline and expose the slender penis extending from the anal region to the urinary orifice. The central tube of the penis is the urethra, the duct that drains the urinary bladder. Cut through the skin slightly to avoid one side of the ventral midline and expose the slender penis extending from the anal region. To expose the remaining structures, cut through the tissues in the midplane between the legs. Spread the legs wide apart as the incision is completed as far as the urethra. The urethra appears to be heavier in the male than in the female, due to the presence of certain accessory glands. The Cowper's glands, about half an inch long and a third as thick, occur laterally and posteriorly, and the prostate gland, oval and about quarter-inch wide, is found dorsal to the junction of the bladder and urethra. Dissect these structures free of the rectum, and expose the point of entrance of the sperm ducts into the urethra at the level of the prostate gland. In the male, the urethra serves for the transport of sperm cells as well as urine to the outside. The operation of the reproductive system. In the mature male, sperm cells are produced in tubules in the testes. Study the demonstrations of the developing sperm cells in a section of mammalian testes and a free spermatozoa of pig and human. As they are produced, sperm cells are discharged into the epididymis, where they ripen over an extended period, after which they are capable of fertilizing an ovum. They are discharged in a seminal fluid or semen, which is composed mostly of secretions from the prostate and the bulbourethral glands. After the seminal fluid is deposited in the vagina of the female, some of the spermatozoa eventually reach the farthest point in the uterine tube, the upper end of the mammalian oviduct, where they are ready to unite with any discharged ova, or egg cells. Ova develop in the ovary in little hollow structures called follicles, beneath the outer wall. Study the demonstrations of ripening ova in sections of ovaries of cat and human. When ripe, the ova burst out of the follicles and into uterine tubes, where fertilization, the union of sperm and ovum, normally occurs. After fertilization, development of the embryo follows in a complex series of events, including the formation of the placenta in the uterus of the mother, where the greater part of embryonic development occurs. The nervous system. Coordination of activities in the mammal is accomplished through two general mechanisms: nervous control and hormonal control. Although their functions may be separated on the basis of mode of action, they are nevertheless interdependent and supplementary. Nervous control depends on direct stimulation of effector organs, the muscles and glands, by nerve cells. Hormonal control depends on the production of an excitatory or inhibitory chemical hormone in one organ, its distribution by the blood to all tissues, and finally, its selective action on a particular organ or tissue different from that which produced it. In brief, a hormone is a substance produced by one tissue that either stimulates or depresses the activity of another tissue elsewhere in the body. The nervous system can be divided into two distinct parts based on function. The voluntary nervous system, under the conscious control of the individual, consists of the neurons of the thinking apparatus, plus those that receive stimuli from the major sense organs, and those that stimulate the effectors, the skeletal voluntary muscles. The second part is the autonomic nervous system, under involuntary control. The autonomic system governs the so-called vegetative activities, such as digestion, secretion, excretion, and circulation. And effector organs are cardiac muscle, smooth muscle, and glands, that is, all the effectors except voluntary muscle. These effector organs are, in addition, subject to hormonal control. The autonomic nervous system is subdivided into two systems, the sympathetic and parasympathetic, both which innervate the internal organs and whose actions oppose one another. In general, the sympathetic nervous system stimulates heart action and depresses the activity of the digestive system and associated organs. Conversely, the parasympathetic, which includes the vagus nerves, tends to slow heart action and promote digestive activity. Many additional functions are known, some of which contribute to the emotions of the individual, through the many connections with the central nervous system, without the person being aware of the reason for or the source of feelings. Likewise, some of the activities of the autonomic system are actuated through the voluntary nervous system. For example, salivation, involuntary, at the sight, smell, or anticipation of food. The voluntary nervous system receives the stimuli of which we are aware, correlates them, and evokes response to them directly through the action of voluntary muscles or indirectly through the autonomic system. The effectors of the autonomic system are the involuntary muscles and glands. The autonomic system is composed of sympathetic and parasympathetic systems, which supply a double and opposing innervation of the internal organs. Hormones affect, among others, structures also under autonomic control. Connections between autonomic and voluntary nervous systems allow the modification of conscious behavior through emotions seated in the autonomic system. The brain. A complete dissection of the brain and 12 cranial nerves will not be made, but enough will be done to give an idea of the major features of the brain. First, remove the rest of the skin from the head and neck of your pig and cut away the external ears. Remove whatever remains of the upper and lower lips. Turn the pig on its side, left side up. Turn your attention to the lower jaw. Pull away the membrane of the dorsal surface so as to expose the milk teeth still embedded in the jaw. Extract one. Note that the cavity of each tooth is filled with a jelly. To expose the brain, remove the roof and side of the skull. Heavy scissors are best to use. Begin at the top of the skull and pick the bones away one small piece at a time. To keep the brain undamaged, free the tough covering of the brain from each bit of bone before it is broken away. The brain coverings are called meninges. The tough outer layer is the dura mater. The finer inner closely following the contours of the brain is the pia mater. An intermediate layer is too thin to see. Several bones will be removed at this point. Now is a good time to identify some of them. Immediately between the orbits is a pair of frontal bones, the cingulate and humen. Behind them are the parietals. Under the frontals and parietals is the cerebrum, consisting of two cerebral hemispheres, the thinking part of the brain, the part that is, with which you cerebrate. Interior to the frontals are the nasal bones, which cover the olfactory organs, parts of the sphenoids, will be picked away in the orbital region, plus the temporal bone behind the orbit, the occipital bones from the back of the skull. To remove the latter, the heavy neck muscles must be scraped free, and the entire dorsal part of the skull exposed. The occipital bones cover the cerebellum, a part of the brain concerned with muscular coordination, and also the medulla, the part from which most of the cranial nerves arise. When the olfactory organ is exposed, make a vertical cut in it. Notice the many folds of tissue, all of which are covered with sensory epithelium. You may also cut into the nostril from above and follow the nasal passage posteriorly both to the olfactory organ and to the pharynx. After the cerebrum, cerebellum, and medulla are exposed on the one side, split the brain into equal halves, left and right, and remove the left half. Now the brainstem, including the medulla, may be observed in its relation to the cerebrum and cerebellum. The pituitary gland may also be found ventrally just behind the optic nerves, in a little pit in the floor of the skull. When you see the location of this gland, you will understand why it is practically impossible to get at it surgically without doing serious damage to nearby structures. The cranial nerves of the pig fetus are hard to find. Attempt to search for them only if you have time.